All right, let's talk about something huge. We're diving into one of the most profound scientific frontiers of our time, the ability to actually edit our own DNA. And look, this isn't just about curing diseases in one person. We're talking about altering the very code of life in a way that can be passed down for generations to come. So let's explore how this even became possible and what it really means for the future of all of us.
I mean, think about that question for a second. For decades, this was pure science fiction, right? But now, it's a very real possibility being worked on in labs all over the world. What would it actually mean to hold the pen, to edit the blueprint of our own species? The implications are just staggering, and it all starts with one powerful idea.
And this is the term for it, heritable genome editing. The key word here, the one you really have to remember, is heritable. See, unlike a normal medical treatment that just affects the patient, these changes become a permanent part of the human gene pool. They are forever. So to really get our heads around this, we need to do a quick refresh on the basics.
Right. Before we can even talk about editing the Book of Life, we've got to understand the language it's written in. So let's start with the blueprint itself, our genetic code.
It all begins with heredity. You know, the simple fact that traits get passed from parents to their kids. It's the reason you have your mom's eyes or your grandfather's sense of humor. All of that information is packed into one incredible molecule, DNA.
Okay, this slide is super helpful. It breaks down a really key distinction. On one side, you've got the genotype. That's the actual genetic code. Think of it like the written recipe for a cake. Then, on the other side, you have the phenotype. That's the thing you can actually see, the observable trait. That's the finished cake. So you've got the recipe versus the result.
So that genetic recipe, it's organized into what we call genes. And different versions of the exact same gene, say one for blue eyes versus one for brown eyes, those are called alleles. It's your total unique mix of these alleles that makes you, well, you.
Okay, so we have the blueprints, the DNA sequence. But it turns out there's a whole other layer of control going on. A set of switches that decide which parts of that blueprint actually get used.
And that's the perfect question to ask right now. Because, just having the recipe isn't quite enough. What if there are notes scribbled in the margins, telling the chef how to use it? This is where the absolutely fascinating field of epigenetics comes into play.
So here's the crucial point. The thing that's kind of mind-bending, epigenetics does not change your DNA sequence. The letters in the recipe stay exactly the same. Instead, it changes how your cells read that sequence. It's like a layer of information sitting on top of the DNA.
And I love this analogy, it's just perfect. Imagine your DNA is a giant cookbook. Well, epigenetic marks are like the sticky notes and highlighter marks left behind by your parents or even your environment that say, hey, use this recipe, or just skip this page. These little notes can turn genes on or off without rewriting a single word.
And one of the most common ways this happens is through a process called DNA methylation. As you can see here, these tiny molecules called methyl groups act like little chemical locks. They attach right to the DNA. This makes that section of the genetic code coil up super tightly, which makes it totally unreadable. The gene is still there, but the cell just can't access it. It's effectively been silenced or turned off.
All right, so we understand the code, and we understand the switches that control it. For years, all scientists could do was read the book. But now we're learning how to rewrite it. Let's dig into how that incredible leap happened.
I mean, that leap from simply observing the genetic code, like you see happening here, to actively changing it, that is one of the biggest jumps in the entire history of biology. It's the difference between reading a map and literally getting to redraw the roads. And a brand new tool made it all possible.
And that brings us to genome editing, the deliberate, precise alteration of DNA. The technology that blew this field wide open is called CRISPR-Cas9. You've probably heard of it. It's become famous because it's so efficient, pretty cheap, and has just stunning accuracy.
Honestly, the easiest way to get your head around CRISPR is to think of the find and replace function you use in a word processor. It gives scientists the power to search for a very specific string of genetic letters and, poof, change them into something else.
So here's how it works in 3 simple steps. First is find. A guide molecule is programmed to hunt down the exact DNA sequence we want to change. Once it finds it, step 2, cut. An enzyme basically acts like a pair of molecular scissors, snip, and cuts the DNA at that precise spot. And finally, replace. The cell's own natural repair crew rushes in to fix the break, and we can give it a new piece of DNA as a template. We basically trick it into replacing the old sequence with our new one. Find, cut, replace. It's just that powerful.
Okay, so now that we know what's possible, we have to face the much harder question.
Just because we can do this, should we? And this is where the conversation moves out of the lab and into, well, the global town square.
To help us navigate these incredibly tricky waters, organizations like the Nuffield Council on Bioethics have brought together experts to really think through the social and ethical issues. Their work gives us a really important framework for this whole debate. And the Council came up with two fundamental principles to guide us. First, any change has to be for the welfare of the future child. That's number one. And second, it has to promote social justice and solidarity, not create new divisions or inequalities in our society.
And these principles force us to confront some massive dilemmas. I mean, what about safety? What are the long-term, unintended consequences of making a permanent change to our DNA? What about social justice? If this is expensive, will it create a genetic divide between the rich and poor? Then there's the social impact. What message does editing out certain conditions send to people who are living with them right now? And maybe the biggest one of all, do we, just this one generation, have the right to make choices that will alter the human gene pool forever?
So, where do we go from here? The Nuffield Council basically recommends hitting the brakes, not the gas pedal. They're calling for a broad public debate right now before this stuff becomes widespread. They want independent oversight, super strict case-by-case licensing if it's ever even permitted, and long-term monitoring to track the consequences for people and for society.
You know, ultimately, this isn't just a question for scientists or ethicists anymore. The power to direct our own evolution is nearly in our hands, and that leaves us with the biggest question of all. Who gets to decide what we become?